Let's start off with photosynthesis. Photosynthesis is a process in plants that converts light energy to chemical energy. Photosynthesis begins with this little guy, a tree, a plant, or a leaf. The first step of photosynthesis is when photons of light from the sun reach the plant. What does the structure of a leaf look like? Well, every leaf contains openings at the bottom of a leaf called stomata. There are also cells known as mesophyll cells. In these mesophyll cells, there are oval-like structures called chloroplasts. In chloroplasts, there are small pancake-like discs called thylakoids. This is where light-dependent reactions occur. A stack of thylakoids is called grana. As different stacks of grana are connected, the chloroplast is formed. Surrounding these stacks is a jelly-like substance called the stroma. This is where the Calvin cycle, or light-independent reaction, occurs. Now let's take a closer look at the thylakoid. The thylakoid has two parts, the thylakoid membrane and the thylakoid space. The thylakoid membrane includes chlorophyll. This is where electrons are excited from photons of light that shine through it. In the chlorophyll, we have two types of photosystems, photosystem 2 and photosystem 1. As a photon of light hits photosystem 1, the electron in the chlorophyll causes the chlorophyll to get excited. The electron then gets excited and then is brought to photosystem 1's primary acceptor. The electron then moves to ferredoxin, down to plastocyanin, and back to photosystem 1's reaction center. ATP is produced from the energy released during this process. This whole process is known as cyclic photophosphorylation. The next process is called non-cyclic photophosphorylation. In this process, an electron is excited and reaches photosystem 2's primary acceptor. Since the electron in this process doesn't return to the reaction center, it is replaced by an electron that's released when water splits. When water is split, this process is known as photolysis. The electron in the primary acceptor then travels to plastoquinone, then to plastocyanin. The electron is then placed into photosystem 1's reaction center. When a photon of light hits the reaction center, the electron is excited and is transferred to ferredoxin. It is then used in the NADP reductase to produce NADPH from NADP and hydrogen. The electron then gets replaced by an electron from photosystem 2, and the process repeats. As photolysis occurs in the thylakoid space, the two hydrogen ions from the water transfer from the thylakoid space to the stroma by traveling through the ATP synthase. As it travels through the ATP synthase, ADP and an inorganic phosphate combine to form ATP in order to power the Calvin cycle. Now let's talk about the Calvin cycle. The Calvin cycle occurs in the stroma and is known as a light-independent reaction. The Calvin cycle produces the final product G3P, which is glyceraldehyde 3-phosphate. The phases in the Calvin cycle are carbon fixation, reduction, and the regeneration of RUBP. In carbon fixation, the stomata open at night and CO2 is taken in and combined with RUBP, or ribulose biphosphate. The enzyme Rubisco catalyzes this step, and then two molecules of 3-phosphoglycerate form. Also, this is powered by ATP and is then broken down into ADP. In reduction, the phosphate from ATP is combined with the 3-phosphoglycerate to form 1-biphosphoglycerate, and then transforms into 6 G3P molecules. This reaction is powered by the reduction of NADPH to NADP and an inorganic phosphate. One molecule of G3P exits the cycle, and the remaining 5 are recycled to regenerate the 3 molecules of RUBP. The last and final step of the Calvin cycle is the regeneration of RUBP. In this phase, the remaining 5 G3P molecules are transformed into 3 molecules of RUBP by the reduction of ATP to ADP, giving G3P's phosphate to RUBP. Since the RUBP is ready to combine with CO2 again, the cycle continues. Now that photosynthesis is complete and has an output of G3P, the glucose molecule is now ready to be used in cellular respiration.
Cellular respiration is an enzymatic breakdown of glucose in the presence of oxygen to produce cellular energy called adenosine triphosphate, ATP for short. The 3 main stages of cellular respiration are glycolysis, Krebs cycle, and electron transport chain. Glycolysis happens in the cytosol/cytoplasm. Glycolysis starts off with the nutrients such as glucose. ATP is used to drop off a phosphate group, and this forms ADP. This process occurs twice. Then the 6-carbon sugar diphosphate molecule is then split into two 3-carbon molecules. Each of the 3-carbon molecules is converted to pyruvate. Electrons are then transferred to the coenzyme NAD+ to form NADH and ATP. The Krebs cycle happens in the matrix. Pyruvate enters the mitochondria and NAD+ produces NADH. The pyruvate splits into acyl CoA and CO2, and then the remaining 2-carbon acyl portion is transferred to a 4-carbon molecule, producing a 6-carbon molecule. This molecule releases CO2, producing a 5-carbon molecule. Hydrogen is removed and taken to the NAD+ to form NADH, and CO2 and NADH are produced again. Finally, a 4-carbon molecule is formed, and then oxidized and removed hydrogens are used to form NADH and FADH2. The reaction regenerates a 4-carbon molecule that reacts with acyl CoA, and then the Krebs cycle repeats again to break down pyruvate. Lastly, the ETC chain happens in the inner mitochondrial membrane. Coenzymes NAD+ and FAD are then reduced to NADH+, H+, and FADH2. Next, electrons from NADH plus H plus are transferred to the electron carrier proteins. The electrons move from cytochrome to cytochrome. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Proteins are also transferred to the outside of the membrane. The cytochrome oxidase complex then transfers electrons from cytochrome C to O2, the terminal electron acceptor, and H2O is a product. The proteins then re-enter the matrix passed through ATP synthase. Protons are used to synthesize ATP from ADP and phosphate. Formation of ATP is called oxidative phosphorylation.